Good morning, students. Today, I'm going to talk about history of microbiology, theory of spontaneous generation, experiments of Pasteur and Tyndall. Basically, this is a course I'm taking on microbiology and industrial application. So this is the first chapter of this course where we are going to talk about the history of microbiology and theory of spontaneous generation. In the 17th century marked the beginning of the revolution era in the science. For thousands of years, humans lived unaware of the teeming microbial world and surrounding them, and disease, decay, fermentations, and several other mysterious processes attributed to supernatural forces, bad airs, are being considered. But what is behind that is not clear, and that is the start of the history of microbiology. The first glimpse of microbes, 1676, the discovery of microscope through Antonie van Leeuwenhoek, a Dutch lane merchant, with an extraordinary curiosity, construct a simple yet powerful microscope. And he had made at least 300 different types of microscopes of different magnification in order to increase the magnification of the small molecules. And later on, he called the living organisms seen through these microscope animalcules, little molecules. Now, the scientific documentation, Van Leeuwenhoek meticulously documented this observation, and he wrote at least 200 detailed letters to the Royal Society of London in order to prove that this is the small molecule called microorganisms. Well, legacy and recognition, his groundbreaking work earned him recognition as the father of microbiology. Now, Leeuwenhoek's revolutionary tool, that is the discovery of microscope in 17th century, unlike the compound microscopes of this time, and his simple microscope consisted of a single perfectly grounded lenses mounted between brass plates. And this instrument called microscope was fitted to check and to see the various samples, to look into the small molecules like animalcule, later they called as the microorganisms. His hand-drawn sketches of animalcules, which was earlier called as small molecules or animalcules, living microorganisms, provided the scientific community with the first visual documentation of bacterial morphology. That is, how does the bacteria looks like, including rod shapes, spherical, and different shape of bacilli, coccus, and different shape of bacteria were observed. Early belief about the spontaneous generation. Now the next question came, and how does these microorganisms grow, and how do these microorganisms appear? In order to know the growth and the appearance of these microorganisms, there are two schools of thought. One is called spontaneous generation, another is called the biogenesis. So there's a two school of thought, there's a tug of war between the scientists and the philosopher of that time, that whether the living things are generated or are grown from the non-living organisms or non-living stuff, or from the living organism. That was a big debate at the time. So there's a classical examples like ancient observers believe maggots spontaneously generated from the rotting meat. Mice arose from the dirty rag, and wheat and eel emerged from the mud. So these were the concept, and these seemingly logically conclusion were based on the casual observation without controlled experiment. So all these things are established without any evidence. Now the microscopic extension helped this to understand. Even after Antonie van Leeuwenhoek's discovery of microorganisms, many scientists extended spontaneous generation theory to the microscopic world, and they argued that the bacteria and other microorganisms, they arose spontaneously without any living things from the organic matter, mud, even air and stuff. Now the scientific authority, based on the evidences, they try to prove that the organisms are not produced from the spontaneous generation, but they should have some background living organism, that is biogenesis. And the theory of persistence stemmed from this endorsement by respected authorities like Aristotle, later scientist. Now he demonstrated that how scientific paradigm can become entrenched, requiring extraordinary evidences and persistence of challenges to overcome. Now, Francesco Redi, 1668, performed an experiment. The first controlled experiment was performed. Francesco Redi, an Italian physician and naturalist, educated one of the first controlled biological experiment in history. Now, his experiment was he took an open jar, flies could access the meat freely. Then he covered the jar and fine cloth with a fine cloth and prevented fly access while allowing the air to circulate. And third experiment, he took a control, identical conditions except for the single variable. Now, revolutionary results came out of this experiment that maggot appeared only in the open jar where flies could lay egg, definitely providing that maggots came from egg, not spontaneous generation from meat. So this was the first experiment to disprove the spontaneous generation and to prove that maggot produced from the biogenesis. Well, the battle against spontaneous generation and the biogenesis going on, and the microscopic debate continues. Now, there are three experiments was performed. John Needham in 1745, he was an English naturalist, scientist who heated Newton broth, that is a broth for the microorganisms, and then sealed them, these flasks, and then microorganisms appeared. He concluded that arose spontaneously, arguing that this heating had killed all the pre-existing life. But Lazzaro Spallanzani in 1768, an Italian priest and scientist who boiled the broth for extended period and sealed flask completely. Now, after boiling, is basically kill all the earlier microorganisms or life in the flask, and then no microbial growth were found, which was challenging Needham's conclusion and supporting the idea that microbes came from outside sources, not spontaneously. Now, the question of air. So, critics argued in Spallanzani's extended boiling had destroyed the vital forces in air necessary for the spontaneous generation, and keeping the debate alive and highlighting the need more sophisticated experimental approaches. So, this debate continues on, and one group continuously supporting the spontaneous generation and other group continuously disproving the spontaneous generation and try to prove with experimental evidences the biogenesis. Now, the Louis Pasteur's swan-neck flask experiment in 1861 was a great experiment to disprove all the spontaneous generation theory. Now, what he did, he took a flask, prepared the Pasteur's filled with a flask with the Newton broth, and drew out their neck into the long, like swan neck-like structures and curved S-shaped, resembling swan neck. And this unique design allowed air to enter while creating a trap of a dust particle and microorganisms. So that way, all the air is not distorted, but they came out from outside into the flask and they trap into the long S-shaped neck. The sterilization process, he boiled the broth thoroughly to kill any existing microorganisms and then allowing the flask to cool. And the curved neck permitted air exchange, addressing critics concerned about vital forces destructions. So he again disproved the vital forces, which was earlier talked about, who might destruct the life. The observation period during this experiment, the broth remained sterile indefinitely time, and then providing that the microorganisms did not appear. And so he proved that microorganisms did not appear spontaneously. Now, this experiment definitely disproved the spontaneous theory or spontaneous generation of microorganisms. So establishing that life comes from life. So this was the concept who tried to prove by the Louis Pasteur. Now, the Louis Pasteur's swan neck flask experiment stands as one of the most beautiful examples of the scientific methodology in the history, which is totally evidence-based experiment. And the curved neck serves as a perfect filter, allowing air to enter while preventing the contamination, but addressing every criticism of the previous experiment. Now, the last Pasteur's legacy beyond the spontaneous generation. Pasteur's contribution to the microfield of microbiology is great. Now, coining the microbiology word, the Pasteur's coined the term microbiology. and established it as a distinct scientific discipline, as a whole, discipline as a, biological sciences. His work demonstrated that microorganisms play a critical role in fermentation, disease, changing the life, decompositions, and various benefits of the microorganisms and the harm effect. Pasteurization development is also by Pasteur's, time. And he developed a pasteurization process, using controlled heat to kill the harmful microorganisms from the milk, wine, and beer without destroying their nutrition value. So that make the food stuff, good quality and free of all microbial growth. Fermentation is another, and disease is another area where the Pasteurs play a very important role. The Pasteurs proved that specific microorganisms cause fermentation and disease, establishing the germ theory of life, germ theory of diseases. His work showed that many diseases previously attributed to miasma or bad air were actually caused by the specific pathogen or specific microorganisms, not by miasma or bad air. Now, John Tyndall's fractional sterilization in 1876 was a great breakthrough for sterilization. During sterilization, it was found that many spores which are heat tolerant, which can stand at the high temperature of boiling and high temperature of sterilization, can withstand and can remain, and after some time they regrow and produce the microorganisms or their life. So what the Tyndallization process is, John Tyndall, what he did, he boiled the medium to kill the vegetative bacteria, allowing cool to permit spore germination, boil again to kill new germinated bacteria, and repeat this cycle couple of times, 3 to 4 times, to complete the sterilization, so that the spore which are re-emerged after some time may again produce the maggot or may produce the microorganisms, and then they are all heated and killed. Now, this method, known as the fractional sterilization or Tyndallization. So, Tyndallization is the ultimate mode of sterilizing the microorganisms from various, the foodstuffs, pharma industries, and other organizations it has been used. Now, what is the germ theory of diseases? This is again breakthrough in the history of microbiology. Now, as the earlier thought, the miasma to germ, changing the disease theory. So it is not the miasma but the germs which are responsible for causing the disease. Now, ancient theories were early civilization attributed the diseases to angry god, evil spirits, imbalanced bodily humor. The treatment focused on the appeasement, purification, ritual, or attempting to restore the humoral balance through bloodletting and purging and so on. That was the early concept. Now, miasma theory, that is the bad air theory, dominating medical thinking because disease like cholera, plague, were thought to spread through noxious vapor from swamp, sewer, and rotting matters. That may have some link even today's science. But the Groloma Francesco proposed a new concept, that the contagium vivum, called living contagion, in 1546, suggesting invisible living agent, that is the microorganisms, which cannot see by the naked eye, cause diseases. Ignaz Semmelweis, another philosopher, demonstrated the hand washings importance in the preventing the child fever in the 1840s. Now, the modern understanding of the microorganism, that Pasteur and Koch postulates, work established that the specific microorganisms causing a specific disease leading to a targeted prevention and treatment strategy that formed the foundation of a modern medicine. That was a breakthrough in the microbiology or microbial history. The Joseph Lister antiseptic surgery was established in 1867 because at the time the post-surgical infection was not known how it happened. Even today, the post-surgical infections are very common. Now, at that time, the surgery was performed through not keeping things in sterilized mode, and the pre-Lister surgery was conducted in the dirty conditions with the unsterilized instrument. Surgeons wore their bloodiest coat as badge of the honor, and post-operative infections is quite possible. Carbolic acid solution was used by pastors who were inspired by the Lister, began using the carbolic acid as a phenol to sterilize the surgical instrument before surgery. And he also developed a carbolic acid spray to purify the air during the operation in the theater. The dramatic results were found the post-Lister's antiseptic method, which reduced the post-surgical mortality, because post-surgical mortality has become very common because of the infection caused after the surgery. So having the sterilization mode after the surgery, the infection can be controlled and the mortality rate can be reduced by 50% or less than 15%. His success proved that microorganisms cause surgical infections and that preventing contamination could save the lives after the surgery. Robert Koch, considered as the father of medical microbiology, his contribution in the field of microbiology is tremendous. Koch also proposed called the Koch postulate for infection establishment. Koch's major achievements are first to prove the specific bacterium caused the specific disease. So anthrax in 1876 was identified by the Robert Koch, and he considered that is caused by a single individual organisms, not by something else. He developed a solid culture media using gelatin and later agar-agar, which is very helpful today, invented techniques for isolating pure bacterial culture, and identified the causative agents of tuberculosis in 1882 and cholera in 1883. That was again a breakthrough in the field of microbiology. Received Nobel Prize in Physiology and Medicine in 1905 based on the contribution towards the microbiology. Koch's work provided the experimental proof that Pasteur's germ theory needed transforming the medicine from empirical practice to evidence-based science. So these are the Koch postulate. As I told earlier, there are four postulates with Robert Koch has proposed to establish the disease germ theory to support the germ theory. The consistent association, the suspected pathogen microorganisms must be found in all cases of the diseases. So it means you can identify the microorganism from a specific disease. So specific microorganisms causing a specific disease can be identified. Now these microorganisms can be cultured. So isolation and purification of the microorganism from the disease model can be possible. You can culture it into the in vitro condition. Then third, this experimental reproduction, that is, you can again, using these microorganisms, you can again use the susceptible healthy animal model or human model to again produce the disease of the same kind in that animal model to prove that this animal is causing disease caused by the same microorganisms. Now again, you can re-isolate the same microorganisms from the animal model where you have developed a disease. So basically, these are the four Koch postulates are establish the germ theory of diseases. Now, what is the impact of the Koch? We should know as a student of microbiology in the area of medicine, or especially in the microbiology or clinical microbiology. Tuberculosis discovery is the Koch postulate, is the Koch's impact. Koch identified the Microbacterium tuberculosis in 1882, providing the infectious nature of TB and enabling the development of a diagnostic test and eventual treatment method on TB. He also developed a cholera, investigation of a cholera. His work in Egypt and India led to the identification of Vibrio cholera. Then he also targeted the treatment method by identifying the specific pathogen. Robert Koch work enabled the development of a targeted antimicrobial treatment rather than the general therapeutic agents. He also worked on a vaccine development. His postulate guide vaccine development by ensuring the researchers targeting correct pathogens for immunization strategy. So identifying the correct pathogens, develop the vaccine against that, and the concept of the vaccine is again by the Robert Koch. Several laboratory methods were developed. Koch developed a solid culture media, staining techniques, photomicrography, then establishing the modern bacteriology and laboratory. So all these contribution of the Robert Koch in the area of microbiology make the field very established and good. Now, the golden age of microbiology. Now, the Pasteurs and Koch were the two contemporary scientists and also rival. The rivalry and collaborations also helped the microbiology to grow. Late in 19th century witnessed an unprecedented period of the microbiology discovery, largely driven by the parallel but occasionally competitive work of Louis Pasteur in France and Robert Koch in Germany. Their different approaches and national rivalries spurred remarkable scientific advancement, both by Robert Koch and Louis Pasteur. Some other pioneer microbiologists at that time, which contributed a lot to the field of microbiology, like Paul Ehrlich, 1854 to 1950. He was a German physician who developed a chemotherapy and concept of magic bullet. So the concept of a drug designing or concept of the drug is basically started from the Paul Ehrlich concept. Then Ignaz Semmelweis, who was in 1818 to 1865, is a Hungarian physician for demonstrating the hand washing dramatically reducing the fever we call the purple fevers, which is causing the death. At the time of pregnancy, this kind of a fevers develop and mother might die because of this puerperal fever. His work placed germ theory but provided crucial evidences for disease transmission prevention. So see, it is in early 19th and mid 19th century concept now is fulfilled and is valid even today, as WHO has already proposed the word wash, clean, hygienic wash, that is water with clean and hygienic condition. The Frances Nightingale in 1820 to 1910, a British nurse who revolutionized the hospital sanitation, that is a hospital management system in nursing practice. Her statistical analysis proved that poor sanitation or sanitary conditions cause more military death than battle wounds, leading to major healthcare reform. That was a big concept which is now quite valid even today's concept. The microbiology's expanding horizon and several new concepts in the area of microbiology has emerged as the new microbes were discovered and new microbes associated with the different diseases, different infections discovered. For example, viral diseases, discovery of the viral infections. So Dmitry Ivanovsky work on the tobacco mosaic disease in 1892, which was a plant virus, led to the discovery of the virus. An infection agent is smaller than the bacteria. So that was the first discovery in 1892, explored as even smaller than bacteria, there is some organisms called virus. Then come to the fungal infections. Fungal infection is again, other than the bacterial infections in medical mycology, is a big discovery, a big invention, which is causing the various pathogenic fungal diseases, causing diseases like ringworm, thrush, and systemic mycosis, and several other fungal infections might cause the deadly diseases. Then discovery of the protozoa, we call it protozoology. The protozoology again was the area where people came to know about the diseases caused by various protozoans, not by bacteria, viruses, fungus, but another microorganisms called protozoa, called malaria parasites, plasmodium, then sleeping sickness, trypanosoma, then linking vector-borne diseases to specific pathogens. Then the development of immunological breakthroughs, where the understanding of immune responses of the host, antibody formations against the pathogens which infected, acquired the immunity development, then leading to improve the vaccines. So this concept of immunological breakthrough bring some new concept of vaccine development, the immune system of the host against the viral, against the bacterial fungal infections during the disease establishment. Modern microbiology building on the past, the molecular revolutions, then DNA sequencing, then we have it revolutionized the microbial classifications and identification, revealing the evolutionary relationship, invisible through traditional methods. Then antimicrobial therapy. Now, the antimicrobial therapy is basically even today is emerging area where that due to drug resistance, the antimicrobial agents or the antibiotics are failed, and these antibiotics are no more be useful in order to cure the diseases or subside the infection. Therefore, the resistance can be cope up with the development of a new antimicrobial discovery. So, to discover the new antimicrobial agents, the concept came from the Fleming's penicillin discovery, which has saved millions of the lives and evolved the new response to resistance challenges. Then vaccine innovations. Modern vaccines from the recombinant DNA vaccines, mRNA vaccines, even the vaccines, earlier vaccines we call vaccines which is attenuated vaccines, are the whole cell vaccines which can protect or which can prevent from the various diseases. So, Pasteur's pioneering work while incorporating cutting-edge molecular techniques, how the vaccines can prevent the diseases. So, that's all come under the modern microbiology. The microbial research basically help also to understand the whole genome profile of the microorganisms, and that can also help through genomics and proteomics, the new concept of genomics and proteomics, to understand the disease target, both designing the drug molecule as well as to understand the the insight of the microbial response against the host. So, host-parasite interaction can easily be understood through these genome or the microbial research. Why microbiology is matters today, right? The pandemic preparedness, antimicrobial resistance, then biotechnology applications, environmental solution. Now, this is very important to understand the coming pandemics. As earlier in 2019-20, we have a COVID-19 pandemic, which took away a lot of lives, millions of lives globally, and that is because of the infection caused by SARS-CoV-2 virus. Now, how to prepare these pandemics is again very important, and in order to understand this, it is very important to learn about the microbial biology, whether it is a microbiology or vaccine, or it is a virus biology or fungal biology. But I think this is important to understand what kind of pathogens are circulating all around. Accordingly, you have to be prepared about in order to prepare either through vaccine development or through hospital management. The resistance is another issue. It's a growing threat of antimicrobial resistance bacteria requires continued innovation in the drug development. So you have to again cope up with this also. How can we prevent from the emergence of the drug resistance and provide the new antibiotics to the patients having infection caused by the several deadly drug resistant bacteria. Environmental solutions is again, microbes play a very crucial role in bioremediation, waste treatment, carbon cycling, harnessing microbial processes, help address environmental challenges and climate changes. So all these things are to be taken into consideration to understand the whole holistic view of the microbial world. Biotechnology applications, microorganisms serve as factories for the production of the medicines, biofuel, and industrial chemicals. Understanding microbiology about the genetics of microbes, metabolism, enable sustainable biotechnology solutions. How can we produce that? Now, this is end of the talk, and I think next of the lectures will be started from in the next lectures we will discuss about the more of biotechnology applications and continuing some other part of this topic.